My name is Peter Mulligan, and it's my pleasure to introduce our next panel, which is titled Modeling and Protection of Dynamic Marine Ecosystems. This panel is going to highlight our current understanding of the science and the technology used to study the dynamic ocean. And after taking into account the migratory behavior of many marine species, it's going to offer its insights to policymakers for identifying areas for conservation and to better protect ocean species. Our first panelist is Salvador Jorgensen. He is a research scientist at the Monterey Bay Aquarium, where he studies the ecology, migration, and population dynamics of pelagic fishes, including great white sharks. He got his PhD at UC Davis, studying the population level implications of fish movement. And today he's going to speak to us on the conservation of marine apex predators, accounting for the spatial and temporal distribution. Our second speaker is going to be Ben Lascelles. He is the coordinator for the identification and designation of important bird areas at BirdLife International in Cambridge, UK. In addition to coordinating the conservation of over 300 bird species worldwide, Ben represents BirdLife at international groups such as the High Seas Alliance and the UN World Ocean Assessment. His talk will discuss seabird data as a tool for marine conservation. And our final speaker is Daniel Costa. He is a professor of ecology and evolutionary biology at the University of California, Santa Cruz, where, among other things, he studies the movement patterns of highly migratory marine mammals. He is the co-founder of the Tagging of Pacific Predators program, which has studied the migration of 23 species. And he will talk about the spatial management of highly migratory marine species and the importance of population viability. So just a reminder, we'll have 1 or 2 questions immediately after the speakers, and then we'll have a session for questions later. And so now I'll introduce Salvador Jorgensen to come give the first talk.
Thanks, Peter, and thanks for the organizers for putting this on. It's a great opportunity to come and speak about my work and hear all the interesting diversity of work from all these fields that I'm unfamiliar with, economy and policy. And having this all come together, we have kind of these similar goals, and I think it's a great event. So thank you. And thanks for sticking around for the last session. Our group has tried to replace acronyms with glossy pictures of marine megafauna, so just mix it up a little bit here. And my goal in this session is to try and illustrate some of the challenges of managing highly migratory species, particularly sharks, as in the face of sort of the emerging science of what we know about their spatial dynamics and their particular life history. And I'll be using white sharks as a case study, and I'll be exploring that a little bit. Then we're going to move into multi-species and finally ecosystem level ideas. And white sharks are particularly timely, I think, case study, as currently they're under consideration for maximum protection in the United States under the Endangered Species Act. This is a single species approach, and there, to my knowledge, has not been a highly migratory ocean species or fish or shark that has been listed. And but interestingly, the white sharks are not alone right now. There are 2 other species currently under consideration. And I think these proposed listings draw attention to an issue that's garnered increasing attention, which is conservation concern for sharks worldwide, and this has been brought up here today. Sharks are particularly vulnerable to overexploitation due to their slow life history traits. They're late to mature and they have few offspring. And there's a huge demand for sharks currently for the shark fin soup industry. So I'm going to talk about white sharks, and as I go through this case example, I think some of the challenges that I'm referring to will become very obvious. And I should note that a lot of what we're learning about white sharks is turning out to be true for many of the different highly migratory large predator sharks that ply the open oceans. So white sharks were once considered to be a coastal species when we knew very little about them. But it turns out that they actually spend more than half their lives out in the open ocean. They move. We tag sharks here along the California coast, and these sharks move thousands of miles out into the open ocean. But they don't move out randomly and wander around in this apparently featureless environment. They go to specific locations and have very regular timetables. And in fact, they return back to local spots here along the California coast with great regularity and over long time periods. And what we're learning is, despite their great capacity for movement, and they do move on ocean basin scales, their return in natal phylopatry is causing genetic isolation into smaller subgroups. And these are opportunities for management, for assessment, but also to some extent make these species a little more vulnerable. Now, it was only really with the advent of satellite tagging some 10 years ago that we discovered, as I mentioned, that white sharks actually utilize a much broader habitat than just the coastal areas. And the sharks pictured here, these are a running tally of satellite tracks that we have through the Tagging of Pacific Pelagics program, also known as Tagging of Pacific Predators now. And these are basically like a breadcrumb trail. These are daily estimates of position. And what you see, these sharks were all tagged in this region of Central California. And they move off into the ocean, but their movements are, as I said, not random or evenly spread out. They're concentrated in particular areas. So there are three core areas: this coastal area along the coast. There's this area out near Hawaii where a lot of these sharks go, and that extends out throughout the island chain all the way. Here's the Big Island, and here's Midway Island out here. And then we have this big area where sharks tend to go out in the Pacific. In this region, some people have noted as the desert of the ocean because of this sort of lack of productivity in this area. And we just call that the White Shark Cafe. And I'll get into why we call it the cafe a little bit later in the talk. But the gist of it is that while trying to understand the importance of this area, migration is a costly endeavor. And species that migrate long distances usually have two things in mind. Either they're going to eat or they're going to reproduce. And a cafe may be somewhere where you could initiate either of those activities. And since we're not sure exactly what they're doing out there, we've dubbed it the White Shark Cafe. So this spatial pattern also has a temporal component. We see sharks here on our coast in the fall. And with the onset of winter, these sharks tend to move out. And by June and July, virtually all the sharks, and these are adults, I should point out, are offshore at the cafe and in the Hawaiian Islands. And then come summer and fall, they begin returning again. And as I mentioned, they return back to the California coast with great regularity and precision. And I like this anecdote because it really illustrates site fidelity in white sharks. These pop-up tags that we use do not relay positions in real time. They collect data while they're attached to the animal, and at a pre-programmed time, they pop up and then relay that information. And it's sort of like Christmas when you're waiting for that tag to pop up and tell you what happened. And in 2005, we tagged a shark not far from here. I think we're about in this area here, San Francisco, out at the Año Nuevo Island at a large elephant seal rookery where the sharks hang out. And that tag popped up 10 months later, 700 yards from where it was placed. So we said, well, that wasn't very exciting until we began looking at the data and we realized that it was this incredible journey out to Hawaii lasting over 8 months and return back to the very same spot where we saw it the year before. And it turns out that this data set is comprised of many of these long return journeys out into the cafe and out into Hawaii. And so this has been a big revelation for us and a very exciting development. The fact that the sharks are returning to the same spots, and we know that these spots are where the major seal, E-seal, elephant seal rookeries are along the Central Coast, Año Nuevo Island, the Farallon Islands, Point Reyes. We employed a second technology, acoustic tagging. Acoustic tags don't collect data, they just emit a ping. And that ping can be detected only when a shark is within range of one of the receivers that we put out. And so knowing that these sharks are going to return to these spots, we began putting these out at these key areas. And we collected a tremendous amount of data and began putting these out in 2006 and have gotten this very regular return, year after year, further reinforcing this site fidelity. So at the same time that we tagged these animals, we also took tissue biopsies and analyzed the DNA, and we realized that these white sharks, although they're found all over the world, actually are composed of these smaller populations that are genetically and demographically isolated. And so we have a northeastern Pacific white shark population. And I think in the parlance of the ESA, that would be was it a distinct population segment, DPS? So we have populations here, and just in the Pacific Ocean, we have 3 different populations. Even though these sharks are clearly able to navigate and swim on transoceanic scales. So now that we knew that this was a sort of a closed population, we knew sort of the boundaries of where this population could be found. We started using a third marker, a tag, if you will. The dorsal fin of a shark is like a thumbprint or a barcode. Each one is unique, and those unique sets of ridges and valleys are kind of like, as I said, a barcode, but they are persistent over long periods of time. And so we began collecting these photos during our field work, and we have a large collection of 600 photos and have been able to identify 170 unique individuals and set up a mark recapture study. And it turns out when we compare some of these photos with historic pictures that we found through our collaborations at some of these locations, it turns out many of these sharks persist and return to these areas over decades. Here's a picture from '93, and you can see it's exactly the same after 16 years. In fact, this fall, I was out there, and we were able to photograph Tom Johnson, who was first identified in 1987. Been coming here for 26 years, and probably is the world's longest studied wild shark. And it's interesting because the estimated life expectancy of a white shark is approximately 30 years. And so this suggests that these sharks, once imprinted to an area, may come there year after year after year for their entire life to one particular hunting hole, one foraging area around an elephant seal colony along the California coast. And so using this information, we were able to try and estimate a population size of northeastern Pacific white sharks, those that are found in Central California. So just to summarize a little bit about this structure within the Northeast Pacific, we have these sharks along the California coast. They're moving off into the Cafe over to Hawaii and back again. There's another group of white sharks that aggregates at another elephant seal rookery at Guadalupe Island. These sharks move off to the same region here in the Cafe, and they return back. It's a very, very similar pattern. So we estimated that there were, on the order of between 200 and 300 adult white sharks in this region. And some similar studies suggest there are around less than 200 adults in this area. There may be other locations in the Eastern Pacific where white sharks aggregate similarly, but those have not really been identified yet. A third component of this, and I should repeat that this, we're talking about adults here, and then adult habitat. Adults feed on elephant seals, and juveniles. Juvenile shark species are typically deposited in nursery areas. Sharks give live birth. It's amazing, and they go to specific designated historical nursery areas, drop off kids. There's no parental care. That's it. They're gone. Good luck. And these are typically coastal areas, mangrove areas, or shallow productive regions. In the Eastern Pacific, that happens to be the Southern California bight. One of the big spots is right off of Malibu Beach, actually. And that area extends from Point Conception through Santa Barbara, Hollywood, and then all the way down into Baja, California. We have fewer data for juveniles, but this is a similar plot to the one I showed you at the beginning. Now, there's an interesting pattern here. So here we have animals moving from these EEZs of USA and Mexico out into the high seas and back. Here we have juveniles living along the coast within the EEZ, and they're moving back and forth across the Mexican border seasonally, and this has to do with temperature. With the onset of winter, these sharks head south, and we have some data now that indicates that they return up into this area as the waters warm up. So juvenile white sharks are exposed to fisheries both in California waters and in Mexican waters. Along the desert coast of Baja, you can regularly find these mummified remains as evidence for their exposure to fisheries. They're not really a targeted species. There's not a lot of them, but they are incidentally caught. So we have this piece here now to add to this picture. So here we have a complex migratory pattern for one species, and it's interesting to note that once these juveniles develop, then presumably they move into these adult habitats. Now, when we compare the movements of white sharks to their closest relatives, also lamnid sharks, the salmon shark, and the mako shark, with similar data. In yellow, we have the white sharks. I showed you these three key areas. Now look at salmon sharks. They're occupying this whole northern region of the Eastern Pacific, and mako sharks further south. So understanding one species is not really enough. We realize that probably over evolutionary time, these species have diverged and have been able to sort of partition out the ocean and minimize competition between them. But there are areas where all three overlap. In this case, this is the California current. And here I'm showing just a simple plot that shows when and where all three species overlap in red. And you can see that this is a seasonal pattern off the coast of California. Now, with the Tagging of Pacific Predators program, we've taken this and utilized not only sharks, but fish species, pinnipeds, whales, birds, etc. And we've brought these all together and tried to put together density maps to show where these regions of highest overlap of species, predator species, are in the Eastern Pacific. And here you can see those areas in relation to the EEZs. And I'm not going to get too far into this because this will be probably in the realm of Dan's talk, but as PI have taught. But I will say that we spent a lot of time looking at correlations between environmental variables and the density of these predator species, both in space and time. And without explaining the graphs, because we're running a little short on time, I'll just say that I was very interested in listening to Alistair's work with correlating tuna abundances with temperature. The big explanatory variables in our data set were indeed temperature and chlorophyll. However, those could only really account for about 20 to 30% of the deviance that we saw within our data. So I think one important point that I want to bring up is some of these, as we look at correlates or indicators of what areas are important in the ocean, we need to really look at, we need to understand individual species because they're each different, but also we need to look at them all together to see where the overlaps are. And as we look at, I think, proxies such as temperature and chlorophyll of where to expect these animals or how this would change in the future under the face of climate change, it's important to note that some of these top predators, just due to their size and mobility, have kind of a buffer between the indicators of primary productivity that we can see from space and their actual presence. And I'll just illustrate that with an example of elephant seals. The elephant seals that Dan studies, they aggregate at those rookeries that I showed on the map, but they go off and feed near Alaska. And they come back to the California current and to molt and to mate. And so it's important to understand what activities are involved with their presence in certain areas. And not only that, white sharks come to visit those rookeries to eat the seals, but where the calories from the seals are coming from the Arctic. And so there's this huge disconnect between where the primary productivity is and where ultimately these larger predators may end up. And I think this is a complexity that we haven't really addressed yet. We're still kind of in the infancy stage, I would say, in looking at correlating environmental variables. So this is a huge time lag, it's a huge space lag, really, and it's a complex problem. So I think I'll end there and hand it over to the next speaker, and I'll be happy to take a few questions in the meantime. Thank you.
We have time for questions.
Run down before I take up any more time. Hi, this is Rebecca Martone with the Center for Ocean Solutions. It's really interesting that you brought up the point of linking this idea of predators and their prey. When you think about this idea of where activities are happening in the ocean and the risk and threats from cumulative human activities to these places and trying to identify hotspots for management, one of the things that you brought up there at the end was thinking about those relationships. So I'm just wondering, in terms of your recommendations for how to do that, or if you want to have that discussion. I just think it's an important point that I just wanted to highlight. Thanks.
Yeah, thanks, Becca. I'm not sure I'm the person to answer that question. I think that it's very difficult. I don't think I can answer that. Sorry. What? You don't have to. Good. Maybe we can address that in the panel a little bit. All right, I think I'll let Ben go. And shit.
Yeah. So do you have an idea if you just split the world ocean into two, EEZ areas and high seas?
Yeah.
How long do they stay in the high seas versus the EEZ? Any ideas at the moment?
Yeah, I think we have a graduate. One of Dan's graduate students is working on that exact problem. I haven't, and I'll tell you for white sharks, which I know, it's about 60% of their time as an adult is in the high seas and 40% approximately in the EEZ. With the juveniles, I would say it's 100% in EEZ and 50% in one country and 50% in the other.
You have a paper?
We have one coming out soon. Do we have a, yeah. All right, thank you.
Great, and here's our next speaker, Ben Lascelles.
Thanks, Peter. Hi, everyone. Afternoon. Thanks for the invitation to be here. So I work with BirdLife International in Cambridge, UK, as part of the global seabird program there. And I'm going to talk about how we might use seabird data as a tool for marine conservation. So first off, why are we interested in seabirds? They make up a group of about 350 species, 4% of the world's birds, but they're actually the most threatened group now. 11% of all critically endangered birds are seabirds. They occur in all parts of the ocean, and they utilize many different resources and occur at many different trophic levels. So they're very useful for acting as an indicator group as to what's going on in the marine environment more generally. And we actually know a lot about seabirds, probably more than any other marine taxa. So I'm going to just break my talk up into three quick sections. The first of these is looking at how we've tried to pool and mobilize all this great seabird data that's available through new databases and apply it from a conservation and kind of policy perspective, which often wasn't what the data was originally collected for. And then I'm going to touch on how we've inputted that data to fisheries management discussions and also into discussions on marine protected areas and marine spatial planning more generally. So developing new databases. Satellite tracking devices, we've already heard a great introduction to that. There's a lot of data out there on seabirds, and BirdLife has been coordinating the compilation of this data into a central database. I'll come on to that in a moment. We've conducted literature reviews of foraging preferences of seabirds to try and define distances that birds travel, particularly during the breeding season, what habitats they associate with, and if there are key prey items. And this information helps us to predict the distribution of birds, but also input to what the management of those areas might require. And we've pulled all this together into a first list of critical sites for seabird conservation globally, which includes 3,000 sites. So I'll show you that at the end. So the Seabird Tracking Database, this is a unique collaboration. BirdLife manages this database on behalf of the global seabird community. We have data contributed on 40 species now on this online database. And this is just a video from the website where you can search by species, breeding site, colony, life history stage. You can view the data online, and you can make data requests to the data owners to use that. So this is one example of one species, Cory Shearwater, which is relatively well studied. So we have contributions from nearly 100 scientists now, lots of data held offline as well, which we're looking to include there, and we've just received some small seed funding to try and expand this beyond the albatrosses and petrels to look at penguins, particularly focused on inputting to the Antarctic marine protected area discussions. So this is the current extent of the data that's available online. We've got a pretty good global coverage there, and fantastic resource for using this to inform ocean basin scale management, but also at the site level. So the tracking data has really formed a core input for us to try and use this to implement changes or encourage changes within fisheries management. And the key focus for us from a seabird perspective is bycatch. Seabird bycatch is the biggest threat to albatrosses and petrels globally, and this occurs primarily in three types of fisheries. Longline fisheries, which can kill 160 to 300,000 seabirds a year. Gillnet fisheries, potentially up to 400,000 seabirds a year, and also in trawl fisheries as well, deaths occurring on the warp cables and at the nets during setting and hauling. So as I said, bycatch is the primary threat for a wide range of albatross and petrel species. Here's a picture of a particularly bad fishing trip that ended up catching a high proportion of birds. When you scale up this kind of catch across a fisheries, you come up with these kind of figures that we're talking about. And there's now data proving that this is behind some huge declines. So this is an example for black-browed albatross in South Georgia, where the colony size has been reduced quite markedly, and bycatch is thought to be the cause for that. So fishery, RFMOs, regional fisheries management organisations, we've particularly focused on inputting data to the tuna fisheries, and they have a duty to conserve species affected by their fisheries, including seabirds, turtles, dolphins and sharks. So for us to achieve some conservation successes and for us to understand where and when bycatch is actually likely to be an issue, this helps us decide where to focus conservation action. And this tracking data has proved to be a vital resource to allow us to do that. So this is the Cory shearwater, again, a species that breeds in Europe, in Spain and Portugal, the Azores and the Mediterranean. And via the tracking data that's been submitted here by more than 10 different researchers, we're able to get an overall picture of the distribution of this species throughout the year. So the underlying data here is the distribution of Cory shearwater, breeding up in the Atlantic, migrating into the South Atlantic, particularly off South Africa during the non-breeding season. And we've overlaid this with ICCAT fishing effort, number of hooks per 5-degree grid cell, and you can see there's potentially some negative interactions going on there, or there's the potential for bycatch to be occurring. And this has really proved to be a useful method for actually getting fisheries organisations to recognise that bycatch is a problem, and also to try and guide what kinds of mitigation measures you might put in place for that. So BirdLife has produced white papers for all of the 5 tuna commissions that overlap seabird distribution and fishing effort to highlight where some of those key areas for interaction might be. And we produced this policy brief for the Kobe Joint Tuna Commission meeting last year. So that's the fisheries inputs. From a marine protected areas perspective, there's lots of buzzwords out there: connectivity, representativity, networks, indicators, ecosystem approaches, and seabirds and seabird data can really form a core component of assessing whether these conditions are being met and help us to track how effective our network of sites might be. So here it is again, the Cory shearwater, and this is the extent of the data in the database for this species. So clearly, ocean basin scale conservation is going to be necessary. And for something like bycatch, that's probably the scales that you might want to tackle things at. But actually, from an MPA type perspective or a marine spatial planning perspective, you need to be able to pull out some of the hotspots within this that are going to be more useful for that kind of site-based conservation approach. Say again?
When is it on the water and when is it in the air by thousands of meters?
Well, so you'll see the migration rate coming up for this species. So this is a different species, the great shearwater, and this breeds in the South Atlantic, and this shows you the distribution of individuals each day. So it breeds on Tristan da Cunha, a UK overseas territory. So activities there, we would like to protect feeding areas, rafting areas near to the colony, some potential for that happening through some kind of MPA. Birds then track across to Argentina and Uruguay before migrating north off the tip of Brazil up to the Bay of Fundy and the Grand Banks. And we use different analysis methods, and here they are, all are coming back down again towards the end of the year. So we use first passage time, kernel density analysis, and area restricted search to define where these hotspots might be. And we do some statistics to try and identify how representative our track birds are of the overall population to allow us to make sort of population level assessments. So I think this illustrates very nicely some of the difficulties of conservation for a migratory marine species. You're looking at potential, doesn't have to be MPAs, but some kind of enhanced management measures needed with this species in mind in these different areas. So you've immediately got the UK and Argentina needing to talk together, which in this day and age is not straightforward. You've got Argentina and Uruguay needing to talk to each other, which again is not necessarily straightforward. You've got different regional seas conventions, you've got different fisheries organisations operating, so how all of these conventions and countries work together, talk together to implement some kind of conservation strategy that's going to be effective for this species is a real challenge. So we've undertaken that kind of analysis for all the data in our seabird tracking database, pulled together information on breeding colony locations, and launched this through this interactive electronic atlas last year at the Convention on Biological Diversity. So you can search by country, and this links through to a description of this particular site. You can see which species it's important for and during which times of year. You can also look at the data from a species perspective. So this is the wandering albatross, and this links through to case studies about threats and actions that you might implement to improve the conservation status of that species. So this gives you an overview of its overall range in the Southern Oceans and some of the critical sites for it. So this is a really great tool. There's 3,000 sites on here. It's had input from probably getting on for 1,000 seabird and marine scientists around the world. We have sites in over 120 countries, country profiles highlighting potential conservation actions that would benefit seabirds there as well. Given our geography, I'll give a plug to the Barrow to Baja work that went into this. This was a collaborative project between National Audubon, Bird Studies Canada, Pro Natura in Mexico, and several other partners. And this was looking to define a network of key sites for seabird conservation from Barrow to Baja. And this shows the map of the 100 or so sites that were identified up in Alaska. And these incorporate over 20 million birds, but in just a quarter of the, or less than a quarter of the actual marine area there. So potential for some real conservation gains for big numbers and diversity of species in relatively small areas. So the data held in this e-atlas and the tracking database more widely we feel should really become essential information for a whole range of sectors, conservation practitioners and policymakers. It should inform energy sector planners, go into environmental impact assessments, be used by fisheries managers to assess bycatch, but also potentially catch limits, particularly in important feeding areas, and it can obviously be used to guide marine pollution management plans. And we've had some really good uptake of this data through a range of regional agreements already, the EU Birds Directive, the Nairobi Convention for the West Indian Ocean. It's been used in Japan to inform offshore wind farm designations, and we've had a significant input to the Convention on Biological Diversity process to identify these EBSAs and gain some good successes there in getting our sites incorporated within to EBSAs. And I think that's because we have a scientifically robust process that's relatively easily explained and has some consistency across large regional or ocean basin scales. So I'll leave it there. Thank you very much, and happy to take any questions.
Do we have time for one questions?
Thank you, Ben. Martin Hall again. I just wanted to say this is one of the issues that we wanted to identify things that work. And at the Commission, the information from BirdLife is received periodically. BirdLife is present at the meetings. Many of the resolutions from the Commission are based on that information. For instance, the extent of mitigation measures is defined by the areas that come from there. Policies or spatial issues coming from the information. And it helps, first of all, the scientific seriousness of BirdLife, and that comes down from John Croxall down, which is somebody we all respect and like. But it is a very effective approach. They are present at all the meetings. They are doing their share, and the information is received. We cannot produce it. We don't have the resources or the ability to do it. But it's a perfect case of a very effective, without too much noise, and just producing something that we don't have. So it's I think it's a great example. We're trying to work with the Inter-American Sea Turtle Convention to produce a turtle equivalent for the Eastern Pacific. But it just shows why the example should be followed.
Well, thank you for your endorsement. Very kind. I think it's true to say that seabird people are probably quite lucky to have us as a conduit, the scientists, to feed that information through to relevant policy processes that we're following. I think a lot of scientists don't necessarily know how or when they can use their data to inform policy opportunities. And perhaps that's where the disconnect lies between that science and policy interface. And you know, if similar things can happen with turtle groups and cetacean groups, it would help us massively to have other examples of how that might work. And we're very happy to share our experiences of how we've approached things with other groups.
Daniel Dunn, Duke University. We see each other a lot at these meetings, and most of the meetings we're sort of required to give them the sort of static boundaries around the areas that we feel are important. And you showed a lot of those today, and I'm curious what steps BirdLife is thinking about taking in terms of actually maybe not just putting forward the boundaries, but putting forward the information that allowed you to build the boundaries so that somebody might be able to use that for the sort of dynamic measures that we were talking about earlier.
Yeah. Well, so lots of the underlying data is available there, either through the tracking database and things, and if people want to utilize that, they can. We can handle all those kind of data requests. What we've done through this process is define sites in a consistent methodology. So we know where the sites are, we don't necessarily know why they're there. And I think some of these habitat modeling processes that people have touched on a bit, you know, ideally we should do that for every one of these 3,000 sites to understand the underlying variables. Some of them are based around seamounts, coastal, shelf break, canyon areas, in which case they're probably relatively static, but a lot of these, particularly more generalist species, might target frontal zones and upwelling regions that could be spatially and temporally dynamic. And I think there's a lot of potential to develop the kind of models that have been done for tuna and things to, you know, the overall site is very large, but where the management needs to occur at any given time might be quite specific. And I think if we can understand those variables better from a site's perspective, I think that's where you can really get some key management gains. But I'd be reluctant to take that on myself with so many sites and such a small team. But you know, I think if we can explain the need and the benefit of approaching things like that, that you might see some of these management bodies wanting to develop that themselves, hopefully.
Our last speaker, Dan Costa.
Yeah, it's really sort of daunting to follow those two talks, but I'm going to try and do my best to sort of step back a little bit, at the same time give you some case studies. And I'd also like to say that the BirdLife is a tremendous example of the two-way street between the research community as one of the teams that's put data into BirdLife. It shows what working as a community and not just a bunch of individuals, how the collective data can be put forward to answer some really important management questions. So it's really fun to come here today to climb down from the ivory tower and talk to people that actually do things and can make things happen. And as you've seen, especially from the previous two talks, we've got a lot of tools, a lot of tremendous opportunities to study these animals in ways that we never did before. Over my own career, I actually worked with one of the pioneers of some of this technology as a postdoc, and so I've seen how our ability to study these animals has changed. And so I want to sort of address that, because one of the things I've seen, and I've been part of this, is we get these amazing data sets, and then we say, oh, let's protect this area. But we have to ask ourselves the question, is this the right place to be protecting? Is protecting this place or this place appropriate? And so I want to sort of step back a bit and ask that question. And I was part of a workshop that David Kaplan and a few people, Daniel Dunn and others, were at that workshop in Sète, France last year. And they sort of asked the question, how do you set up population viability? How do you deal with a highly migratory species? And I'm not going to explain this slide because it would take too long. But what this slide represents development of coastal MPAs along California, where they've taken into account the life history characteristics of different species. An abalone, which doesn't move very far, it has limited larval dispersal, versus a rockfish or a cabezon, which has much larger movement patterns. And they asked the question, how do you set up different size MPAs, and how big does the MPA have to be to keep a viable population also in the presence of fishing? So I use this as a backdrop to say that those of us that in the highly migratory community really haven't embraced this yet, and that was one of the things that we were looking at that workshop. So this is really sort of a launching point that got me thinking about what I was going to talk about today. Now this is meant to sort of, this is the counterpoint to Sal. These are the things that Sal's sharks eat. These are elephant seal tracks, and so these are just showing you some of the data we get, and these are female northern elephant seals from two colonies, from Año Nuevo and from Año Nuevo colony. From Año Nuevo colony, and from down here, and every time I push this thing, it stops. I'll stop doing that. So from Año Nuevo, which is right here, and San Benito. See, these are the two almost the most northern colony and the southernmost colony. And there's a couple of things I want you to see from this. Once there's a migration phase, we'll talk about that. This is the migration phase. They're all feeding out here, whether they're breeding on the island at Año Nuevo, on the mainland island, Año Nuevo or San Benitos. Both groups of breeding areas, both breeding spots are going to the same area of the North Pacific Ocean. Now, this becomes somewhat undaunting if you say, let's protect. Fortunately, nobody's that worried about elephant seals. They're doing fine. But as an animal, to understand the question, trying to deal with management of that entire home range is just not going to happen. If you think that's bad, you already saw some of this from Ben's talk. This is sooty shearwaters, some data we collected as part of the TOP program. The blue shows where they are at their breeding site. So this is a small island off New Zealand. This is where the sooty shearwaters are feeding. And then they migrate, they move this way, and then go across the Pacific. 3 major hotspots off California, Alaska, and sort of Russia and Japan, and then come back to New Zealand to breed. How do you set up an MPA over an entire ocean basin? So the question becomes, how do you set up what areas are important to consider? And so I've showed you elephant seals which had different breeding sites and went to a very common feeding site. We had sooty shearwaters that had a common breeding island and then went to different feeding areas. And then I haven't mentioned, I'll talk a little bit more about, is there are more simple species that are much more constrained in that they feed and they breed in the same general area. And so protecting them is a little bit more straightforward. However, the initial idea, well, let's just protect the whole thing. And I've just pointed out this really is not a very tenable task for especially entire ocean basins. So then you can say, well, we know where they breed or where they feed, so let's protect that area. But again, you have to ask the question, is this the part of the population that's in jeopardy? Is this the component of the population, the life history pattern, that really needs to be protected? And unfortunately, we really haven't developed the models necessary to do that yet. And that's one of the things that I really want to speak to is, well, I'm not going to propose any real models, but this is a research effort that really has to take place in terms of how to determine population viability. Do you protect on the migration corridor? Do you protect the feeding grounds, the breeding grounds? What components of that? But still, if you're talking about the scale at which these animals are operating on, that still is a very large ask. So then the next thing is what Alistair Hobday talked about earlier today, is looking at the features that these animals are using. And I'm sort of trying to pick that out here by saying these animals, even though they have a very large home range, there are very specific oceanographic features within that home range that they tend to use and they tend to focus on and tend to do particular patterns over those ranges, whether it's sea surface temperature, ocean eddies, frontal systems. There's a whole variety of things, and a number of us have been spending a lot of time and energy figuring out what are those features that these animals are using and which ones are important for migration corridors, which ones for breeding habitats. Sal talked about the White Shark Cafe, not knowing if it's a hot spot to find mates or it's a feeding location. Again, finding out what these places are and what their function are starts to allow us to figure out how to protect them and whether they should be the highest priority for protection. So I'm now going to sort of go through a few examples that I'm aware of and been involved with in terms of asking questions about population viability. And I'm going to take two different kinds of examples. One, as I said, there's these species that are more constrained, and I use an example here of California sea lions. I'm going to use some sea lion examples to show you management of populations where breeding and feeding are in a much more constrained area. I've talked a bit about elephant seals, and this slide is really just a comparison slide showing you that this is the home range of female California sea lions. This is the home range of female northern elephant seals, just as a comparator. I'm not going to use those species, but I use this figure as just to give you an idea of the difference in scope. So I'm going to take you to South Australia, and it's an animal that I've spent a fair bit of work on, the Australian sea lion. It's endemic to Australia. It ranges from South Australia over to Western Australia. And it's a very interesting animal in that it has a fairly long breeding cycle. It's got an 18-month breeding cycle. Most other sea lions have a year. It's aseasonal. It breeds. It has a long breeding season. It's about 5 months. It breeds on all these little tiny colonies throughout its range. Some of these colonies are 5 to 20 individuals, some up to 400. And it feeds on the bottom. It's a benthic feeding sea lion, which gets it in trouble with a shark gillnet fishery. And so we'd established that these animals were feeding on the continental shelf and that they were feeding benthically early on. And we had done some other physiological studies. But then the population was declining, and the Australian government was trying to figure out what was going on to this population decline. And it turns out that what's going on, using this tracking technology, other colleagues did a complete survey across the the range of this sea lion. And here you can see the home range of at least one area. This was this area right here. So here's the home range of the sea lions, which were tracked with satellite telemetry. And here's this shark gillnet fishery. And then you can see the overlap between the shark gillnet fishery and the sea lion home range. And you can see there's a fairly pronounced region. Now, the Australian Fisheries Management Agency put together a management plan, and I was asked to review that plan, and the argument became, well, how much bycatch can this population withstand? We're talking about 5,000 individuals, a population, and that's of total population. The bycatch is predominantly females, and they were taking somewhere between 20 and 100 a year, and the big argument was how many allows viable population, because the World Trade Organization, they were trying to get certification to export the shark through the World Trade Organization, and the Department of Tourism and Environment had to certify population was viable and recovering. So the argument became, how much bycatch can you have? Well, the answer to the question is, this is an extremely difficult animal to study because I just told you about the strange life history pattern. We don't know how often a female really reproduces, whether they skip years or not. There's all these little rookeries that you have to go and get 20 individuals and look at. The end of the story is that no bycatch is the only way that you could say that the population is viable and is recovering. Another example, which is the New Zealand sea lion, and this one is islands south of us, of New Zealand. Here's the Auckland Island. And what's interesting about this is there's an interaction between the gillnet fishery, with the arrowtooth squid fishery. This shows the fishery region here and here. This animal has two feeding types. One where it feeds on the bottom, and one where it feeds mesopelagically in the water column. We've studied this animal, and we've shown that benthic foraging is sort of the least preferred, least energetically beneficial strategy. It's harder, and that this mesopelagic foraging is more energetically beneficial. So now we see two different foraging patterns. The one that's most beneficial is the one that's most there's the greatest potential for impact with the fishery. So if you're starting to talk about viability, even though you can probably more easily protect this, the population parameters of animals that are benthic foragers is not going to be the same as the animals that are feeding mesopelagically. Just throw this in. They've proposed a SLED, which is a sea lion exclusion device, instead of a TED, which is a turtle exclusion device. The problem is you might actually be getting rid of bodies. The animals could easily get into that and drown, and then you reduce any bycatch because you don't see it. So the final sort of story I want to bring you up with, it takes this theme about different kinds of foraging areas and gives it to you in a sort of almost the two-point perspectives, work that Hoyt Peckham and colleagues did. And it takes together data on loggerhead sea turtles in the central North Pacific and compares them to sea turtles. These are juvenile loggerhead sea turtles. The nesting beaches are over here in Japan. The juveniles go across the Pacific. Some stay here in the central Pacific. Others go all the way to Baja. Now, what's interesting about this is if you're one of these guys, you cover a lot of area, and you can just, the numbers are all here. You cover a much bigger area. It's cooler water. There's lower prey availability. It's cooler water. These are ectotherms. You don't grow as fast. There's not as much food. You cover more ground. You go over here. There's more food. It's warmer, and you grow faster. Now, there's two very different interactions here. This is a shallow gillnet fishery, and this is an artisanal fishery. And there's been efforts to do both, to deal with both of them. The one is the Sea Turtle Watch effort, where they've looked at, this is along the lines of what Alistair talked about earlier today, so I won't go into a lot of detail, showing that where the loggerheads are, where the sets are, and where the interactions are. So there's a difference in terms of where the predominant interaction between the turtles are and where the turtles tend to be. And so what they've done is produced a sea watch. You can go online, and it says, don't set your drift net, or your long lines in this region. And if you do that, you can see in 2006 they weren't following it quite as much, and there's where the sea turtle bycatch was. 2007, they were doing that. The fishers were going outside of this region, and there was no bycatch. So there are tools to start to use these kinds of information to give us much more tighter idea, instead of covering the whole Pacific, these limited regions. Now, in the Baja region, Hoyt Peckham and Jay Nichols have been working to get the artisanal fishery to set up a much more static MPA, but they've also done a thing where they're working at the local level, talking to the fishers and making them really get excited about sea turtles. Because when they first went there, they said, gee, I don't know what the problem is. We just caught 50 turtles last month. And it's like, yeah, okay, you don't see the problem. You just killed 50 of these guys. So the point is that there's different ways of approaching the problem. This is a population that's growing much more rapidly, but it's also potentially an area where there's a much higher potential impact because of the artisanal fishery. I'll just sort of take this as a jumping off point to say that one of the things about this is it's an area where there's red crab is a primary breeding area. It's also multi-species where you get bluefin tuna, yellowfin tuna, and loggerhead turtles. So if you do create an MPA there, you can start to sort of converge and protect a group of just not one species, but multiple species. And as Sal sort of said earlier, we have the top program where we have identified these very large regions of utilization of a large group of marine vertebrates. And so the challenge is trying to figure out how do we implement these kinds of strategies for these multi-species aggregations. And one work that sort of looked at that was Sarah Maxwell's dissertation work, where she took the air-breathing vertebrates from the top program and just took all of the data we had for those animals and plotted their distributions in terms of where you see the greatest distribution of these 6 species in relationship to existing marine sanctuary boundaries. And surprisingly, the boundaries weren't made for this, but it's what is the nice outcome of this is the boundaries of the national marine sanctuaries as they exist today do cover a lot of the hotspots of these species. So in conclusion, it's important to identify the home range and then the components that are most in need of protection, migration, feeding, and breeding habitats, find the oceanographic features that associate with these different characteristics. Then, of course, I can't say this enough, is we have to develop the population level models that can take these disparate aspects of the population and having differential mortality associated with where they breed and where they feed. And then the question of how you then apply this to multi-species aggregations and MPAs is something we still need to work with. And a question came up that I wasn't going to use this slide because I thought I had too many already, but this is the top data set. These are all the different species. You can't see them, but the different colors represent, these are monthly aggregations. This is Autumn and Harrison's dissertation work. And I'll leave this slide, I'll end on this slide. This shows the timing of the different species. So these are whales, turtles, these are the albatross. And you see moving from high seas to EEZ. And so you can, this is when the animals in the Mexican EEZ, animals in the US EEZ, and versus high seas. So we have looked at the movement patterns of all the different species of the top program and looked at when they are in the jurisdictions of the various countries and when they're in the high seas regions. And I'll leave it at that and see if there's any questions.
We have time for 1 or 2 questions.
I'm Christina Jurdy, and I've been dying to ask you this question. What is the percentage of time that the species spend in various parts of the EEZ and the high seas?
It's in that figure.
Yes. It's hard to read from here.
Well, I hadn't planned on showing, because if that question came up, I could give you Autumn Lynn's dissertation, and it's all, we have a draft manuscript, which we're hoping to get out shortly.
We need a short, sweet answer.
It's there, well, there's like 18 species, and so we could tell you for the 18 species what they are. I can't tell you off the top of my head, but I could look in Autumn Lynn's thesis and tell you.
Tagging and tracking biologists have a bad habit of whacking it out on the table so we can see how big it is. That typically means putting up a map that shows that my animal lives everywhere and it shows the tracks going, all over the ocean. I think that's been a really bad PR move for interacting with, people who want to do spatial management, because their first reaction is, that's horrible, we won't be able to do anything.
Right.
So I think the kind of analyses that focus on the hotspots are really critical. Have you seen that now when you present that data to people who are into spatial management, that they breathe a bit more easily than when you were doing it 5 years ago?
I don't know if they breathe. I know that we've finally got our head wrapped around that issue, and part of it is, as a scientist, you want to go, gee, oh wow, and that's why I use that figure. It's like, wow, isn't that really cool? But I also use that for this purpose to say that's really cool, but that's you can't deal with that. And so we really have, over the last few years, tried to refocus, and that was kind of the point of my talk, was we are refocusing our thoughts on how do you deal with this? And you can't just say, oh, look, here we know these animals aggregate here, and therefore we should protect it, because that's only part of the process. So I don't know how people are reacting, but you're absolutely right.
We'll just take one last question, and then we'll bring all of them down.
Can I just add something?
Oh, sure, yeah.
So certainly from the CBD EPSA process, which I've been quite involved in, we've submitted the prioritised layer of IBAs, which has received a good uptake because it is a hotspot type analysis. During the EPSA process, quite a lot of people have submitted raw tracking data to that and not been at the workshop. And that's meant there's no time to interpret the data. People don't know how to do that. And that's meant that none of the data has been used to end up informing an EPSA description. So clearly in that process over a few days, keeping it simple, keeping it focused geographically has been the way to go.
Hi, Dan. Hi. Larry Crowder again from Costa. So I'm going to address the population viability issue a little bit. I think it may not be as bad as you're worrying about, because people who do population dynamics and don't do tracking and spatial analysis have thought a lot about the population viability for mammals, birds, turtles, and so on, based on the life history and the vulnerability of different life stages and so on. Most of that analysis has done some kind of elasticity analysis that looks at the impact of stage-specific survival or fecundity or something like that. And one of the downsides of that approach is that assumes that you're making a small change in all of those vital rates, when some management interventions may allow large changes at a low cost, and some management interventions may only make a small change at a high cost. Right. And so ultimately, for management purposes, you want to know, how can I influence population growth rate per dollar that I spend? The second observation is that all of this is great stuff, but it's through the lens of animals that are big enough to tag. Yep. And for birds and mammals that become, by the time they fledge or leave their moms, they're close to adult size, that may be workable. But for something like sea turtles or tunas or sharks that have a long ontogeny of animals that do different stages, it might be the animals are getting nailed as juveniles and we don't even know where the hell they are in the ocean. So
I totally agree. And what I would say, I mean, I didn't want to make it sound like we can't do the modeling. It was really that it hasn't been done. You're absolutely right. The life history, the demography, all that stuff's off the shelf. What hasn't been done is putting it into the spatial context of what happens if you protect this area, not that area, and you know where the hazards are. So it's really pulling the elements together that I'm arguing needs to be done.
So if you set the bar, we need to have all the vital rates data for this particular species in this particular place, you're going to be in trouble. But if as a first step, you see, I have a mammal-like organism that reproduces at age 4 and might live to be 30, you're getting close.
Well, that's why I didn't have time to develop the tremendous work that's been done on MPAs, for the coastal MPAs, and they've done that. They've taken those models and said, if I have an animal that does this with these characteristics, this is how big the MPA should be. All I'm saying is we need to take that same approach here.
Yeah, let's bring the whole panel up, and then we can ask questions to the whole panel.
Just to complement his question, I think the map should have a small label saying only reproductive individuals tagged, no individuals of less than this size tagged, for the interpretation. No? That's because of the gaps.
Yeah, I mean, what I also didn't say is I didn't show any juvenile data there, and I mean, as Larry pointed out, understanding the juvenile lifespan is important, and I had actually made a note to say that, but I forgot.
Hi, I'm Ellen Hines from San Francisco State, and we do a lot of habitat and spatial distribution modeling, a lot of GIS nerdy stuff. I think the thing that I'd like to bring up that the layers that we don't have are really the human use layers. And so we've got a lot of good tracking data, and we're beginning to be able to downscale some of the climate models and get a lot of bathymetric data. But in order to really look at spatial planning, for instance, the NOAA's human use layer is very low resolution and very generalized and very hard to work with. And for me, that's one of the things that I get really frustrated about. I know in Australia, I'm always very impressed by the zoning, the complicated zoning, and the human use overseeing that being done and being mapped. And I don't see that happening here, and definitely not. We also work in developing countries where we don't see that. And the other thing the other layer that I get very frustrated, and the other person I really want to see at this table are the physical oceanographers and the biological oceanographers. I mean, there's a whole food chain, and there's a bunch of us that have for years just been modeling the sea surface and I know Dan does a lot more than that, but for species habitat modeling, especially for turtles, cetaceans, and these elephant seals, it would be great if we could incorporate more ecological criteria taken from midwater or stratified oceanographic areas. Thanks.
Sort of your comment about the human component. I mean, Jeff, I think it was Jeff that talked about that earlier. We all admit that's a big missing thing, and when those data are hidden from us or hard to get or not properly taken account of, we all are frustrated by that.
Maybe I'll just add, I mean, I think from the seabird perspective, we actually know which threats are likely to be affecting a particular species overall, but you don't necessarily know whether that threat is occurring at the site-based level because the kind of information you're talking about isn't available. So I think you could still make management recommendations in this area, because this species is here at this time of year, these 5 or 6 threats may be in place, and then you need to determine whether that's occurring in that local context.
Fine-scale models. Can you repeat her comment?
She's saying that for climate change models, we really need to bring this information to the table. And I think it's a good point. I think this illustrates that not only do we need the specific information for each species and the life history stage, but we also need the specific threats, the specific impacts of different activities, and then across the board for all the species. You start to think of it as a sort of an overwhelming amount of information that would go into some type of model with regards to sort of a one-size-fits-all dynamic protection system, and I think it's a huge task. But I also think back 10 years or so when we didn't even know where these species went. And so we've made huge progress. The tags are getting smaller. Larry, we're doing experiments on putting these on ever smaller animals. And so, we've scratched the surface, but there's so much more we need to look at through the types of studies that we're doing. I think in the previous session, they talked about how the science has been presented and the policy sort of lagging in its response. But I think this is an area that, as we scratch the surface, we realize how much more complexity there is and how much more work we have to do. But I think by looking back a little bit, we can see the progress that we've made, and that is accelerating. So I think there's hope here for that.
The other comment, you talked about physical oceanographers and the data. A good point is these animals also collect data on their own environment. So if it's temperature, we have now chlorophyll tag. The simple temperature can get collected on almost any of these tags, and then the more parameters, the larger the tag. Therefore, as Larry points out, the fewer animals you can put it on. But that all of these data have to be used, and the problem with the models is when you're looking over large spatial scales, the resolution of the models falls apart. And so you're stuck, if you're looking at ocean basin scale changes, you're stuck with models that have low precision. You get at much finer scales, you get models that are a little more predictive at smaller scales, but then there's still just a model.
Well, actually Sarah Maxwell, Stanford University, and following on Ellen's point, we actually, Dan and Larry and myself, and a whole suite of other top folks have a paper in review right now where we try and put some of that human use data together from Ben Halpern with the top data to see what some of the overlap is, and that actually leads in really well to my question, which is one of the things we're missing in this paper and in general is the dynamic nature of people. We're talking a lot about the dynamic nature of animals, but not so much people. So a good example is the Hawaii longline fishery, and one of the things that happened with that closure is that those fishermen shifted to other nations. They had less regulation, and basically some analyses say that there were probably more turtles that were caught as a result of that regulation. So I was just curious if you guys have any examples or could speak to that in terms of looking at this transfer effect of effort when we put these regulations in place and we're looking at mobile species. And I know that's really complex and dynamic, and it's one of the things we're hoping to address in our working group through COS on this topic, but I was wondering if you had any thoughts.
Well, one of the things that reduced the bycatch of albatross by getting the long line hooks put in the water sooner, but you ended up with more petrels because the petrels are able to dive deeper. So there was somebody once made a comment, save an albatross, kill a petrel. Yeah. I know that's not true, but there are trade-offs like that, just even in gear changes.
Yeah. We need to start putting electronic tags on people. And to a certain extent, we're doing that with the vessel tracking examples, et cetera. I'm working on a project where we are thinking of putting temperature and depth sensors on some of the gill nets in Southern California to look at and looking at the interaction with juvenile white sharks there. And so maybe there's something there.
Hi, Willow Batista from EDF. So I have two quick questions, one for the whole panel, which is, I know that corridors and between different protected areas are kind of like a hot topic in the terrestrial world right now, and I'm wondering if there are any examples of existing or if there's movement towards corridors between marine protected areas or MPAs that are specifically aimed at migratory paths. And then my second question is for Dr. Costa. I'm just wondering what happened with the sea lions and the gillnet fishery, and you said there couldn't be any bycatch, and then what did they do?
They closed it became, it's a quick answer, I think. Went out to 120 meters, and once you did that, it made the shark fishery non-profitable, and it's basically closed.
Death threats ensued?
I don't know. Ask Alistair, he lives there. Well, not in South Australia, but yeah.
Maybe I'll say something on the corridor perspective. I don't know of any, well, certainly for seabirds, there's not any corridor migration route MPAs for seabirds. I'd be surprised if there were any for cetaceans or sharks as well. Maybe there is, but I think one of the difficulties is how you define what this corridor is. What kind of density of movement do you need, and over what time period, and all that kind of stuff to actually define a corridor. If it's in relation to a bathymetric feature, that might be easier, but often it's caused by wind or currents, and it's very hard to link it to a feature like that.
But Cory Shearwater's had that. There was a little corridor.
Sure, yeah. But so we proposed that as an EBSA to the government of Brazil, and they rejected it. So
it's feasible. It just hasn't happened yet.
Yeah. I would add to that and say that in the marine realm, I think we can take a lot of cues for the research that's been done in terrestrial systems in terms of protected areas. But there are some key differences, and I think this corridor concept is one, because corridors on terrestrial systems are continuous habitat where there are resources and shelter, et cetera. In the marine environment, for highly migratory species, there's kind of open spaces. But I think the equivalent in the marine system is distance. Distance between protected areas are essentially the corridor, I would say. And so a lot of work has been done with marine protected areas along the coast, using the distance and spacing between reserves as an important metric so that propagules from one reserve may reach the other one. So I think it's a different system, but it's something that distance has been given a lot of thought in that process.
Hi, I'm Catherine Kilduff, and I work for the Center for Biological Diversity. And we're mostly attorneys in the Oceans program. And I want to thank you for all the scientific work that you do, because clearly you've devoted your life to it. And it's really important. And I work a lot with TURN, Turtle Island Restoration Network, Todd Steiner and Terry Shore here, and also Oceana. And the National Marine Fisheries Service says that they're going to designate critical habitat for the loggerhead sea turtle in water areas. And this is both in the Atlantic and the Pacific. So it was really interesting to see the pictures and talk about what's kind of a adverse modification in the case of critical habitat, but basically a jeopardy analysis for certain life stages, and compare those two hotspots. So I probably should talk to you more about that afterwards, because I think that when you're comparing threats and comparing importance of spots, it leads to the temptation to say that this area might not be as important as this area, and so maybe it's less or more deserving of protection when there are certain species which just need protection to avoid extinction. So if you have any comments on that, I'd be interested. Then I'm also concerned because, like Alistair mentioned, that managers would say, this is just hot spots that we have to protect now. And it seems to me that that's almost going back down the road of single species management. And when we think about fisheries, you think about MSY and how to maximize the yield from fisheries. And why is reducing conservation areas based on individual species a prudent step forward in ocean management when, like Larry Crowder mentioned, that there are animals we can't tag easily? By reducing the areas that we protect, aren't we just going to fish until the next species is diminished? Like a shark that's not as charismatic as the great white shark, but still has some of the life history characteristics that makes it vulnerable. So I was wondering if you could answer that.
I said it wasn't prudent. I said it was manageable. I said the point was, if you, and it's also Alistair made the point, when people look at protecting an entire home range of a species, a group of species, they say you can't do that. What I'm proposing is a way of making it feasible. I'm not saying it's a good, and that's why I then said the rest of my talk was you have to show that this is viable for population survival. And part of that, when I showed the two different strategies, was not to say one was better than the other, but if you used a one-size-fits-all and said, if you modeled loggerhead sea turtles with the growth parameters for the animals from Baja, you get the wrong data because the Central Pacific aren't growing as fast, and the time from hatching to returning back to the rookery to breed is going to be probably twice as long as the time for the animals in Baja. So all I can do is tell you what the data say and how to interpret them in terms of population viability. And that's why I say coming to a group like this is important, because I can give you the biological outcomes. I'm not necessarily prepared to argue in a different forum that this is prudent or not, because I wouldn't use that word in terms of interpreting these data. I wouldn't say that's a prudent outcome. I would say you'd have to protect both of them. But I see your point, is somebody could then take those data and make that argument. I wouldn't agree with that, but it could be done.
It depends slightly as well, I think, on what you mean by protection, or whether you're talking about management in general. I mean, something like bycatch, management of that is occurring across a large spatial scale. So you haven't reduced the size of that, and management at that scale is needed and is effective for mitigating against that threat. But if you were trying to inform pollution, oil spill management plans or something, you can actually say that, if an oil spill occurs within 30 kilometers of a major seabird colony, you're likely to be having a population level effect, whereas 200 miles out to sea, potentially not. So I think it helps make that distinction about the likely impact and the population level impacts that might have. And if you can make it as a population level impact, then that's likely to gain more support and more likelihood of success from a management perspective, I would have thought, through policy mechanisms.
I'm Eric Gilman with Hawaii Pacific University. I've been working with data-deficient fisheries and conducting ecological risk assessments. How would you use information on life history, characteristics, and an understanding of thresholds where you might compromise the viability of a population to understand the relative risks of taxa that are affected by a fishery, so across taxa? When might a change in gear or fishing methods or closure of an area be prudent to protect elasmobranchs at a cost of increased mortality of seabirds, for example?
Never. Well, you know, I think that's why we need to start to integrate all these different taxon groups together, and you know, we're starting to get that kind of hotspot understanding to see where those trade-offs might be needed. I don't think we probably even know that at a global scale or at a regional scale when those things might be occurring. Maybe at specific sites you could do that. And
let me answer that in a slightly different way. I mean, you just pointed out a number of the really critical issues, and there's not an easy answer for them. And what you're talking about is different sets of data taken in isolation, not being used in an integrated way. And I'll just put a pitch in for what we tried to accomplish, and I think we did accomplish in the TOP program. It came about because there was a couple of us, Barbara Block and myself, who recognized we asked similar questions on completely different organisms. We didn't go to the same meetings. She went to the Fisheries Society meetings. I went to the marine mammal or the seabird meetings. What we were able to do in TOP is bring seabird biologists, shark biologists, fish biologists, turtle biologists, marine mammal biologists, all into the same realm. So we were talking to one another, so the kinds of points that you made would become more obvious. And I don't know any other way other than to have forums where we speak to people across disciplines to not get into the single species failures where you study one group of organisms in isolation of everything else.
Next question.
Hugh Possingham, University of Queensland. You just talked about talking to people across disciplines, and I think Ellen's already, a couple of people made the points about money and the importance of people in conservation planning. I mean, the whole field of conservation planning has over the last 10 years, more or less rejected the hotspots notion because it excludes people, forgets about actions, ignores costs, and ignores the fundamental principle of complementarity. So I'm wondering whether, you know, pointing out the dangers of these hotspots maps, there is a set of tools, a whole heap of tools that integrates all this information quite comfortably. Sonation, MarkSan, that builds marine protected area maps, builds zoning maps. It's being used by most of the NGOs. And I think almost all examples of the use of these tools have proven that the big data gap is not biological. It's all social and political and money. And so I think that's the key. I mean, we need maps of what people do, as Ellen said. And that's the data gap. We could collect a lot of data on a lot more species, and almost every published analysis proves that makes very little difference to our reserve system. Sorry, that wasn't a question.
No.
I have to respond to that. Yeah. Sorry, but I mean, I'm from the Coast Guard, Phil McGillivray, and we absolutely need hotspot maps. I'm dealing with oil development in the Arctic right now, and we have to have those to respond to events. And I don't think there's anybody who worked on Deepwater Horizon, which I certainly did in this room, who doesn't know that we didn't have the information that we needed for all of the species that were there. And there was a great deal of confusion as a result of that. And that data still doesn't exist for a lot of things. It took superhuman efforts on many people's parts, but I think Dan is probably aware that we didn't have any idea that the sperm whales never left the site. And if certain of us, like the one talking right now, didn't jump into action and get some money thrown at trying to answer that question, where were they? Were they exposed or not exposed? We wouldn't even have the answer. So knowing that that was a hotspot area for sperm whales, which we learned after the fact, was critical. So there is a role for hotspots. And I would also say that the other role that I deal with in Alaska is that those biological hotspots have to be overlaid with cultural hotspots for the native peoples. And you have to have both of those. So it may not be everything for conservation, but if you look at conservation in the broader sense of saving things from natural disasters like tsunamis or human disasters, which we will have more of invariably, there's a role for that in a context of response to disasters. Sorry.
Other questions? Yeah.
Thank you. Talking to fishermen about hotspots, very frequently the hotspots for the megafauna are the hotspots for fishing. So you close the hotspot and you close 90% of the fishing, so good luck with enforcement. I think the issue is to look for the breaks, to look for bycatch to catch ratios, which is the measure I use. Where can you close with the high impact on bycatch and the least impact on the fishery? If the hotspot is a concentration of bycatch with not so much fishing, then fantastic. If it's not, then you have to work around that. So that's, I think the fishing production of the hotspot should be included if you want to have compliance, because it's very easy to close, then you have to make it stick, really. And the other issue is, every time you close an area, you push effort to other areas. And one of the things that the scientists don't have is the apples and oranges problem that we've been mentioning for years with Larry and other people. How many albatrosses, leatherback turtle worth, and these kind of things. No, a juvenile this versus 5 adults of that. Those questions, there's no scientific answer right now. So the idea of where to put a marine protected area requires a very, very careful ecological, demographic view. What is gained, what is lost? We've been trying circle hooks to reduce sea turtle mortality. We do reduce sea turtle mortality a lot, but they are olive ridleys, which are growing very fast in the Eastern Pacific. And we are increasing the catch of silky sharks, which are declining rapidly in the Pacific. So you cannot suggest the circle hook flat. You have to say, in this country, the consequences may be worse. The losses might be bigger than the gains. So each case you have to look at in detail. And sometimes the progress is faster than the analysis, I think.
Well, I'd just say I don't think any of us said all these hotspots should be no-take zones as a start point. I think from a seabird perspective, that's probably going to be quite a rarity. But what you do want to see is best practice management of all activities there that take into account those species that are found in the area so that you minimise whatever those impacts might be. And if it's still having a population level impact, then maybe you look at no-take zones as...
Just the comment is, you're the first one that's mentioned that if that turtle hotspot, that's not my data, I'm just presenting somebody else's results and efforts, that it would also protect the yellowfin and bluefin tuna. You're the first one that pointed out that. We saw it all along and we kind of go, yeah, I know, isn't that cool? It's a matter of perspective. Those of us in the top program are really into tunas and turtles, and so we thought that was all right. But your point is well taken, and it really is depending on which side you're on, and that's also why I made the arguments about to do what you're saying is how can you come up with strategies where you can sort of get some in between and not just blanket closures.
So, Meg Caldwell, I'm going to bail Pete out here in two ways. First, I'm going to say that Martin and Rashid and John should sit at the same table tonight, because you guys could probably talk about bioeconomic modeling and the degree to which that could really assist in assessing the trade-offs that you're referring to, where you have the biological data and you have whatever social data is available to you, and fleet dynamic modeling, for example, which is what we used in California to help inform the design of our network of marine protected areas. Not everyone understood the bioeconomic modeling, but when it was explained to some of us got a sense of what some of those trade-offs were for species, but also for humans, and what we would be doing to fishing effort and displacement of fishing effort. And finally, I just want to say that it is 5 o'clock, and the faculty club is expecting us at 5:15. And so I know that the chef will be very interested in making sure that we are there to eat his food. So, Pete, if you want to go ahead and start the applause for this panel, I think we can wrap it up for the day.